Okay, so without further ado, let's move on to our next speaker, who is Josh Quick. Josh is a UKRI Future Leaders Fellow at the University of Birmingham, and he has been responsible for the creation and maintenance of the very, very commonly used ARTIC sequencing protocol. So when you hear about ARTIC sequencing, you have him to thank or blame, and he is going to give an overview of how genome sequencing works, the various approaches, technological approaches in the lab for generating particularly amplicon sequencing. Josh, are you able to share your presentation? Perfect. Thank you very much. Yeah, I can see that. Over to you.
Great, thank you. So I'm going to try to give you quite a broad overview of sequencing using the ARTIC protocol and the sort of design, or the sort of the considerations you might want to take into account when you're actually deciding, you know, if this is the right method for you. So so obviously sequencing SARS-CoV-2, it's an RNA virus. This is similar to a lot of other projects that ARTIC have worked on in the past, ARTIC, Zika. We've basically been maintaining and improving, updating this protocol over time. But there are lots of things that you might want to consider when you are trying to decide what the protocol that is best for you, including how long it takes, the cost, how difficult it is to do in the lab, what the risk of contamination there might be, and quite often that will sort of depend on how many separate PCR steps there are. Portability, so sort of how much heavy or large bulky equipment does it use, and then of course number of samples. There are some methods which might be suitable for a sort of small number of samples, 1 or 2, but then that's totally different proposition than if you're trying to sequence sort of 3,000 samples a run, like Andrew was just talking about. So there are lots of things to consider when you're trying to decide what to do, and there's certainly not one good protocol which is better than all the others, or approaches that are better than all the others, given that there are so many different considerations which you need to take into account. So the first sort of question I want to look at is the sort of broad sort of approach, the sort of modality of the sequencing, if you like. And there are lots of these. For example, direct RNA is possible with nanopore, so that sequencing native RNA by ligating adapter onto RNA strands, single-strand RNA using a splint adapter. So that's a protocol which has been developed by Oxford Nanopore. Because it requires you to splint the adapter onto the DNA for ligation, it means that it's best suited to poly-A RNA, which not all viruses are, and even if they are, then fragmentation means you're more likely to enrich for the 3' end of the virus if you do that. So quite often when people are doing that, they will polyadenylate the RNA using an enzyme to create sort of an artificial poly-A on the 3' end of the RNA that you can then add the adapter to. Then there's RNA-seq. There's a couple of different RNA-seq methods that are commonly used. One's called CRISPR, one's called SMART-seq. They can, again, be customized for either poly-A RNA, messenger RNA, or random primed. Then there's probe capture methods. And these last 3, moving away from sort of metagenomic methods into targeted methods now. So the targeted methods would include bait capture, where you try to design probes which will cover your genome. The probes are very long, which means that they have a high tolerance of mismatches compared to amplicon and qPCR. And then usually once you've enriched your library using bait capture, you then combine it with a sort of standard library prep approach. Amplicon sequencing, which is where you're enriching for specific regions of the genome, targeting amplicons. And the way that we do this is by targeting around 100 amplicons for a viral genome, depending on the length, which overlap with each other, such that you can reconstruct the entire genome sequence once you've sequenced the whole amplicon pool. And also, I've added on here qPCR, which isn't a sequencing method, but it sort of shows you for reference. You're only targeting generally sort of 1 to 3 targets using diagnostic qRT-PCR. So tends to have very, very high sensitivity due to the sort of sensitivity of the reaction itself, but also the fact that you don't require the entire genome to be present to be able to detect the targets that you have. So if you put those sort of, this is a bit of a cartoon really, more than anything, but just to illustrate that all of these methods don't have the same sensitivity. The highest sensitivity would be the diagnostic qPCR for the reasons I just mentioned. And then following that, you have the sort of targeted methods, amplicon and bait capture, which might work up to sort of CT sort of 33 maybe, and CT 26, 27, something like that, depending on, you know, other factors. And then at the lower end of the scale, you've got your metagenomic methods like RNA-seq and direct RNA. Direct RNA particularly is quite poor for this because it requires so much input, and also is untargeted. So that's the sort of overview of the sequencing, the sort of broad sequencing approaches you might take, split into the 2 main classes of targeted and untargeted. And then you want to think about, in addition to that, you need to think about your sequencing approach. And then this is sort of how you sort of funnel down into your, you know, the ideal method. So you've got long reads and short read sequencing approaches. Long reads are your nanopore and PacBio sequencing platforms that can generate reads for nanopore anywhere from 200 base pairs all the way up to sort of mega bases, millions of base pairs in single reads. And then you've got Illumina short reads, and these can be, and ion torrent as well, or proton reads. And these can typically be paired end for Illumina, single end for proton. And the read length varies by platform and kit, but typically the longest paired end run mode for Illumina is sort of 2 by 250, 2 by 300, something around there. It doesn't scale up very much more than that. And for proton, somewhere around 200. So you need to make sure that your library is able to be sequenced either with an overlapping paired end mode for amplicon sequencing, or completely cover the amplicon using a single end. So this sort of defines the fragment length, if you like, and obviously to do that you might need to add in some additional fragmentation. So if you start with an amplicon size that's too long for your run mode, for your sequencing run mode, you then might need to optionally add in a fragmentation into the library prep. So we don't do that for the nanopore prep. We target with the ARTIC protocol 400 base per amplicons, which is compatible both with nanopore and Illumina. So it's on the short end of the nanopore ideal range, and it's on the long end of the Illumina ideal range. But it means that you can sequence those amplicons on both platforms without doing any additional fragmentation steps, which we like. And it gives you good quality sequencing data without the requirement to do too much downstream bioinformatics. So the way that the overall ARTIC protocol works is it uses, as I said before, amplicon sequencing. So you make cDNA from your RNA. You add this to two multiplex PCR reactions, which you thermocycle separately. And then you combine them together to generate the amplicon pool. That amplicon pool is then end repaired and A-tailed. A-tailing is where you use a sort of Taq-like polymerase to add on a 3' A tail so that you can then ligate a barcode onto that with a TA ligation. Obviously, it also performs 5' phosphorylation for ligation, and then we do this ligation using T4 DNA ligase, and you get effectively a barcoded amplicon pool. And you can do this 12, 48, 96 different barcodes on a plate before you can pull them together to create your sequencing, your multiplex sequencing library. So you've got two stages of multiplexing here. You've got multiplexing going on in the PCR, you've got multiplexing going on at the sample level, and that gives you sort of thousands of amplicons in the same sequencing library. And really the challenge of designing a good sample prep for viral genome sequencing is really this ability to multiplex at both the target and the sample level in the simplest possible way. And that's actually, you know, quite challenging, and that's one of the things that, you know, I work on all the time. Oh yeah, this is animated, which I forgot to do. There we go. Okay, so there's a few other notable methods that are worth thinking about. So instead of ligation library preparation, there's also quite a strong argument for doing PCR barcoding. So this is a similar approach where you would take your amplicon pools, and then you would do you would use tailed primers to then add a tail, and then a further PCR to add on the barcoded adapters. And this is sort of a two-step PCR, so it works quite well. But obviously, there's the added risk of contamination in the second PCR, because usually in a one-way system, you would start with your clean area, your pre-PCR area, where you prepare your master mix, and you have sort of you keep your template away from that, so that when you're setting up your PCR reactions, you don't risk any post-PCR contameration going into the PCR. But obviously that's difficult if you're doing a two-step PCR, because you don't have to do a PCR. You have to do your round one PCR, and then you have to add some proportion of that product to your second round PCR. So that it's difficult to maintain a one-way system when you're doing a PCR barcoding approach. But otherwise it can scale quite nicely. You've got tagmentation-based library preps. So I've cut a few examples here. These are all based on ARTIC amplicons, but then using sort of Nextera style library preps for preparing them for Illumina. So you've got COVID-19 Corona here from Andrew's group in Quadrum, Mini XT from the group in Northern Ireland. And you basically tagment your amplicons using the transpososome complex, which gives you some adapters to PCR off using barcoding PCR primers. So that's quite a rapid way of doing it. For those of you that have been in the lab, you'll know that tagmentation-based library preps are quite a lot quicker than mechanical fragmentation NPrep ligation-based library preps, because it all happens in one reaction, really. The downside of that being that you actually end up with a fragmented amplicon, which means that you don't necessarily see a primer binding site. You don't necessarily see the primer sites on each end of the read, which can make sort of primer trimming and alignment preparation a bit more tricky. And then a couple of new methods which have come out during the last couple of years. We've got this RC-PCR approach from a company called Nijmegen in the Netherlands, and this is quite an interesting idea to do. As I was saying, the sort of the sort of 2 rounds of multiplexing in one reaction, where you have a pool of probes which provide you the sort of target multiplexing, and they're actually sort of reverse complement to what you would normally use in a your normal PCR primers, and they have a sort of common sequence with the Illumina indexing barcoded PCR primers, and obviously, because they have this sort of region of complementarity here, and you can extend your Illumina primer to contain the sort of gene-specific target sequence from your probe, and this is blocked in the reverse direction, so it just happens in the forward direction to create a sort of multiplexing primer pool inside the reaction. So that's quite an interesting idea, and has been used. In this UK sort of wastewater surveillance project, among other things. And then this one's sort of more future looking. Nanopore have recently announced the that there will be topo cloning based library prep kits coming soon. And we've been testing this in Birmingham. I think we're probably the only ones that have tested it, to be honest. And the idea really is it's based on topo cloning that was that uses topoisomerase to create. It's designed for rapid cloning originally, but because the idea of sort of ligating an insert and a vector together is sort of analogous to adding a sequencing adapter to your DNA templates, you can modify this idea to work with sequencing libraries, and you just have to. The only requirements are that you need blunt dephosphorylated amplicons. So we're working with ONT trying to improve this, but the early signs are that this is quite a good idea, and because the topo adapter can have a barcode sequencing within it, you can do effectively, you could probably do a 12 or a 48 or a 96 plex library just by adding, having one, a topo adapter per barcode that you can add in, and this is fairly fast, about half an hour, sort of less than an hour to do this. So we're quite enthused about this idea. So this is the first paper that describes the ARTIC workflow in its sort of most recent, almost current form. And this was originally developed for doing Zika virus genome sequencing in Brazil. And for that, we had to create the primer panels to sequence the Zika virus. And to do that, we had to develop our own primer design tool because there wasn't anything that was really suitable at the time, even though we did test these things. And this is still in active development. Andy Smith, who works for Nick and me, sort of jointly developed this. He's supposed to be speaking after me, but I don't think he's able to. So I quickly tried to cover how this works now. So the website is available at primalscheme.com, and you upload your viral genome sequence, and this can either be a single reference sequence, it can be a multi FASTA reference sequence, which encompass the sort of diversity of the viral genomes that you would wish to cover with the amplicon scheme, and in the most recent form, an N-masked reference, which is a single sequence where you mask out the diversity. So the coordinate system is the same as your sort of reference genome, but you mask out the bases that you don't want the primers to occur at. And that's sort of becoming our favored approach because it sort of internally works a lot more easily and quickly. So you upload the reference genome, you set your amplicon size and scheme name, and then the software will search it digests the genome into k-mers and searches for those that are thermodynamically suitable, and then sort of positionally suitable in terms of pairing them up, and overlaps. And then it also checks for to reduce interactions. It tries to eliminate pairs which interact with each other during the pool. So the reason you can see here now, the reason why there's two pools, this is the top above the line and below the line. These have to be separated into two reactions because otherwise because they have an overlapping region, they would create a short overlap product in the reaction if they're mixed together. So they have to be separate and run in thermocycled in two reactions. It works on conventional PCR, but multiplex PCR. So instead of having one pair of primers, which will either the forward or the complement sequence for your target, we actually have sort of around 100 pairs in total, so 200 primers, but those are split into two reactions, so about 100 oligos per reaction. And in order to make that work, you have to, as I described, try to make sure that they don't interact, but also to make sure that we use them at very low concentration, so about 50 nanomole per oligo, so that much lower, such that the entire concentration of all the oligos together is sort of roughly what you would use in a normal PCR reaction. And if you run that, you get something which looks like this. This is MinIRN data for Zika, and this is unnormalized, so you can see the difference in amplification efficiency of the different primer pairs from nothing to sort of very efficient. You can see the overlap regions where they have a higher coverage because it sort of combines the two neighboring amplicons coverage. And then this is what it looks like if you normalize it down to a sort of a reasonable coverage, sort of 100 or 200x, depending on what you want to analyze. So let's talk a little bit about what's happened with SARS-CoV-2, because it certainly led to quite a lot more sort of focused development on this protocol and the primer schemes. The first SARS-CoV-2 genome was released in January. Well, this version, actually, if you look down here somewhere, was accessioned on the 17th of January. Within 5 days, we'd been able to fork our Ebola sequencing protocol from the ARTIC protocols.io site and create a new fork, which was specific to SARS-CoV-2, and that's both the primer scheme itself and then the accompanying protocol, lab protocol, to work with it. Over the months, we released some changes to the primers. Sort of wish that I'd use minor versioning now, because we were already up to version 4.1, or we're already up to version 4. But we changed the primers over time to work with the emergence of new variants. We didn't really need a change for alpha, but there started to be significant dropouts associated with gamma, and then more prominently with delta, and then definitely with Omicron. So we had to make changes to the primers. We improved the actual workflow of the actual ligation workflow with John Tyson in Vancouver, and managed to get the sort of ligation efficiency, or the double barcoded efficiency, up to about 75%, and reduce the cost as well by changing some of the reagents out for sort of cheaper versions. And this is a plot here just showing sort of different run configurations using sort of up to 96 barcodes, plus minus washes, which you may or may not use, which you can use with nanopore, but you may or may not choose to, and then obviously the different flow cell scales. But it's possible to achieve about £10 a sample using 96 barcodes, sort of here with no washing, which I think is quite a reasonable approach, if a bit slow, because you do have to generate the sort of maximum run yield from that flow cell of sort of 10 million, 20 million reads, which will take you sort of quite a long time. So if you wanted to generate the data faster, you might want to look at a Promethean flow cell, or fewer samples on a MinIRN flow cell, and running that for a shorter time. And another important thing that has come out during the pandemic, and this comes from a group in Japan, a study led by Itakawa, where they discovered that the significant cause of primer dimers in ARTIC PCR was a specific type of heterodimer, which is called a 3' anchored heterodimer, which is where your 3' sequence exactly matches another oligo. And it doesn't appear to matter so much about sort of interactions which aren't 3' anchored, which I think is quite an interesting discovery. If you look, all of these significant dimer products have a R3' anchored, and if they're not, if you see this sort of overhang here, the complement strand is 3' anchored. And initially we implemented a way to avoid these products, and we made version 4 primers. But then more recently, we've realized that exact matches aren't enough to prevent this phenomenon completely. So we were still seeing these sort of inexact these heterodimers where there was a mismatch, for example, here, here, and here, causing some further issues. So we're now sensitive to detecting these as well, and I think that's going to be potentially the sort of interactions solved, if you like, which we've been working on for a long time. So that'll be great. The other problem is that over time, the diversity amongst SARS-CoV-2 has ballooned, and even if you're it's becoming increasingly difficult to make a scheme which can sequence everything that's previously been seen all the way back to the original reference genome. So this is just an example of the sort of canonical mutations in some of these key lineages. Especially, you can see the spike region is just getting incredibly dense with mutations, and you lead yourself. It starts to make you start having to think: do we need to be able to sequence sort of all these historical lineages, for example, if you were just going to bring one out of the freezer, or should we be focusing just on the current ones to make life easier when it comes to designing primers, or make sort of or how well they're able to work. And I think we're able to do this with all of these lineages at the moment without any gaps, but it's getting towards the time where we might have to think about doing something else. And then I just quickly knocked this slide up a minute before the talk because to cover some of what Andrew was saying. But we used to try to, and this is a dogma within PCR that's been around for a long time, which is that sort of the ideal primer length is about 18 base pairs for PCR. And what we've done here is we've modeled the length of primers which have the suitable TM for multiplex PCR, which is actually 60 to sort of think here we've got a 59.5 up to 62.5. That's the sort of allowable range that we use for doing multiplex PCR. Very, very tightly designed TMs to allow them to work in the same reaction. And then if you look at the sizes of these are just randomly generated Kmers, and then they're colored by their GC content. So you can see that the higher GC content obviously is shorter because 3 hydrogen bonds per base, and then the lower GC require longer. If you consider the sort of local context of the GC within your genome, that's going to change within this range. So we've now made it so that we don't apply any dogma in terms of ideal primer length anymore. So we just say that anything with these TMs is acceptable of any length. So we've become sort of TM fundamentalists in that way. And we now basically allow anything from 18 to 34 bases to be included, which allows you to cover this sort of big range of local contexts of GC within your genome. And you can see that the change that's made between version 3 and version 4, version 4 and the most recent sort of the most recent commit of primal scheme, which is where we used to have this bias towards the shorter primers. We now have no bias because we consider everything to be equal now. And I think that's going to improve things further. Okay, how much, am I run out of time yet?
No, carry on. Okay.
So I want to talk a little bit about the problems that we saw when Omicron was detected. It's characterized like other VOX as having a very large number of mutations, particularly in the spike protein. Let's just show a region of the genome here where you see sort of 4 cluster mutations here, and sort of 8 other mutations here, and all of multiple hits to the primer binding sequences. Sorry, I don't know if there's what that background noise is, and these will cause the primers to fail because we've got these, we've got these 2-3 mutations here on the left primer towards the 3' end, which we know is important for stability and amplification efficiency. And we've got 3 mutations here, which within the binding site here, which are going to reduce the annealing efficiency on the reverse primer. So we know, this is going to cause dropouts, which it does because you can see we've got no sequence here, and we had to rapidly address this so that we could see this is an important part of the S gene. It contains sort of 3 mutations that people are interested in monitoring. So Andy made a new mode for primal scheme, which we've not had before, which is called the repair mode. And what the repair mode does is it compares the primer sequences that you're in the current scheme against an updated reference genome. And if they no longer exist in that updated reference genome, then they're replaced with new primers. So it attempts to repair the scheme without having to design the whole thing from scratch again. And that's, I think, quite useful going forward, because generally speaking, it's easier to order a few patch oligos and replace a few rather than revalidating the entire scheme, where if you do that, you have to sort of address any potential bad primers or interactions which you haven't detected, and solve those problems. So repair, I think, is definitely the future, and it's the first time that we've used that with ARTIC version 4.1, and so we released 4.1. I can't remember how long ago now. It's more than a month ago, but it's just sort of filtering through now, and this is validation data against a panel of 12 variants shown in Rampart here, and you can see that we have no dropouts for any of the 12 the panel of 12 variants, although there are some interesting features here relating to a few lineages, B1.214, which causes this dropout here, and this one up here, which is in P1. And those are actually, if you investigating those in a little more detail, you can see that those are genuine. They do have mismatches to those references, to those lineages. We just didn't include those in the training data, those mutations, which is why it's led to this. So it all looks as expected. And I just wanted to quickly say we have been able to partner with a few companies to make and distribute primer pools for doing this work. Notably New England Biolabs, IDT, and Sigma, and it just wouldn't have been possible without having a large oligo manufacturer working with us, because this is a popular protocol with hundreds of labs using this. If every single person had to order oligos, it would be a large amount of work for individual labs, with much more variability. And sort of duplicate synthesis going on. Having a company which can produce this in large quantities and sell it relatively cheaply has been really useful for this, and I think successful. And then lastly, before I finish, I just want to quickly talk about the sort of portable lab aspect of it. And before SARS-CoV-2, this was an important element of the work that we did. We haven't been able to do any field trips in the last 2 years, but this is the ARTIC mobile lab setup that we developed, and it's a 3-cabinet system which packs up into these bags. This taken in INRB in Kinshasa. This photo. All very neat because it was packed up by Ian Goodfellow, who's absolutely OCD about packing, all labelled. And the way that this works is you have 3 tables with isolation cabinets all in one room. So this isn't generally considered to be the ideal approach to have pre and post PCR in the same room, but we're saying that you can do this in a small area, provided you have the additional containment of these separate cabinets, and you're very disciplined about the work that you do. So we have a separate cabinet for master mix, one for sample extraction and sample addition. That's sort of like low titer template, and then you've got one for sort of high copy post PCR. And each of these has its own set of labware, and there's no cross transfer of any labware, which could lead to amplicon transfer, and even the lab coats are all separate. And we found that this can work. So we have a kit list on the website which describes how this works. These are actually made out of hydroponics sort of growing tents on their side, and they have a foil lining on the inside which helps to reflect the UV light for sterilization. And they also have sort of a few holes which you can use for putting in wires, or you can run the labware from batteries as well. And this is a photo here, cool photo, me, Nick and Ian with the discoverer of Ebola there, JJ Mwembe, which is quite a cool memory to have. And then very lastly, I just want to show that we have a a piece of software called Rampart, which is specifically designed for monitoring amplicon sequencing runs, as you can see here, the sort of characteristic amplicon profile, but also allows you to easily sort of detect dropouts, problems with contamination, problems with read length or coverage monitoring, all at the same time as the run's going on. So this happens in real time. So it just takes the whatever the most recently written fast Q file was and just analyzes it. So that's quite a helpful sort of visual representation of an ongoing amplicon run. Okay, so that's it. Just want to thank all of the ARTIC network collaborators and all of our SARS-CoV-2 collaborators and COG, which I haven't actually listed in the slide deck. But yeah, thank you for listening. And I'll take any questions if anyone's got any.
Brilliant, thank you. There are a couple of questions in the chat. That was a really detailed presentation, and I guess the important thing for you all watching is that these choices about sequencing method, whether that's how you enrich for the virus you're interested in, or the sequencing platform that you use, have an impact. They have an impact on the data, and part of the idea of this course is to think about that end-to-end process. So you can't do bioinformatics without understanding, perhaps not in a huge level of detail, but understanding basically what is going on in the lab. And that's one reason why it's very important, even if you're only work on data and on computers, why this is a really a useful thing to understand. So yeah, we've got a couple of questions. The first question is at Li Ching. Thanks for your presentation. What's the direction for sequencing viruses' genomes? Are there any new technologies that will merge after the COVID-19 pandemic? So I guess the question really, as well for me, is, is there an ultimate, plan here? Is there an ultimate protocol that is going to become adopted going forward?
I think that the ARTIC V1 method, if you like, with doing ligation barcoding, if it's done properly, is quite a good protocol, in the sense that it generates very good quality data and genomes if done properly. But I think that it's quite laborious still, even with the optimizations which we've brought in. And I would really like to see a faster method which had less reagents that we used, that could still generate high quality genomes. What that is, I don't necessarily know. I think that we at our within the group, we're always looking at any new sort of method which could be used. That's one of the reasons that I described the RC-PCR approach, which is I think very strong for Illumina, but less strong for Nanopore, due to the fact that you sort of can create an entire Illumina library within that single reaction. But you can't do that for Nanopore, obviously, because the adapters that you use for Nanopore are DNA protein complexes and can't be added during PCR. So yes, I think that's a very good approach for Illumina, not so much for Nanopore. The topo, I think, is probably best for Nanopore, but at the moment we're seeing that not all amplicons are not all amplicons are ligated as efficiently as each other. So you see some coverage bias, and that's one of the things we're working on at the moment to minimize. But certainly I think that could be an approach going forward. It only generates single barcoding, so we need to work on how we can do the best possible, we can handle single barcoding as well as good as possible. And then of course, the untargeted methods like SmartSeq. So within the group, one of our Brazilian collaborators, Ingra, has got a protocol which is a very rapid protocol for doing random prime SmartSeq, which calls Smart9N for Nanopore. And I think that's using sort of going forward the untargeted protocols, as the sort of, you know, instrument yields go up, that also you know, becomes more you know, more favorable. So I don't know, but I think Nick and I both, you know, would both like to see a new generation of protocol in the next, you know, in the next years.
Yeah, definitely. And that actually relates to a previous question earlier from Gustavo, which is, have you done metagenomics and have you found any concomitant pathogens with SARS-CoV-2? We haven't really done metagenomics on SARS-CoV-2 because of the reasons that Josh illustrated in the presentation, which is it really only works well at very high viral copy numbers and or low CTs. But we would like to, wouldn't we? Because metagenomics is a much more flexible technique in the sense of being able to target multiple pathogens at the same time, and also without introducing some of these targeting biases that Josh mentioned with amplicons. But the sensitivity has not been adequate and the cost has not been low enough to really let us do, you know, this scale of genomes that we're seeing at the moment, you know, hundreds of thousands to millions of genomes. So there are a couple of questions about the bioinformatics pipeline that I think we'll keep for after Sam Wilkinson's presentation. There's two questions that are relevant to Josh's presentation though. The first one was, can you show an example of masked reference file for primal scheme? Is that something you can pick up in the chat maybe, or send a link or something like that?
Yeah, I mean, I definitely have examples that I can share, but I guess the interesting question there is, how do you make them? And that's something that we're working on at the moment. So we should have a protocol that we can put up on the GitHub soon. We've got a student, MSci student called Chris, working on that. But yeah, we're intending to have a pipeline that will take the existing multi-FASTA reference and then output the new format.
Okay, yeah, that would be great. And so another question from Artem was, what number of reads, either nanopore or Illumina, would be sufficient for SARS genome assembly?
If you, for amplicon sequencing, it depends on the, for reference, you know, for reference-based consensus calling, it sort of depends on the sort of the difference between the sort of highest and the lowest amplicon, but it tends to work out around 50 to 100,000 reads.
Okay, so that's the kind of ballpark. Okay, so the questions from Gilherme and Charles will pick up later in the bioinformatics section.